Good morning, students. Hope you all are fine and safe at home. We were discussing about eukaryotic cell and we have read cell membrane, cell wall. Now we are going to study about cytoplasm. Now what is cytoplasm? As the name suggests, cyto means cell and plasm means fluid like. That is fluid inside the cell enclosed by plasma membrane, or you can also say that part of cell present between the nucleus and cell membrane. Term cytoplasm was given by Strasburger, and specifically the word is used for the fluid that provides living properties to the cell. Huxley referred protoplasm as physical basis of life. Term ground plasm, hyaloplasm, cytosol are interchangeably used with the cytoplasm, but actually the liquid matrix of cytoplasm except the organelles is termed as cytosol. That is, cytoplasm minus cell organelles is termed as cytosol. The liquid portion of cytoplasm is termed as cytosol. It's the place where many chemical reactions occur within the cell. The part of cytoplasm containing cell organelles and non-living inclusion is termed as trophoplasm. Now what exactly is cytoplasm? It's basically everything within the cell excluding the nucleus. Inside cell minus nucleus is equals to cytoplasm. It is made up of three components. They are membrane enclosed organelles or the cell organelles, cytosols, inclusion. We have already discussed about cytosol, that's it's the liquid matrix of cytoplasm except organelle. Now we are going to discuss about inclusions. These are stored nutrients, crystals, secretory products and pigment granules. Actually what happens? Protoplasm as you know provides the suitable medium for vital and metabolic activities. These activities result in the formation and synthesis of many substances in the cell. And these substances are actually in the form of stored nutrients, crystals, secretory products and pigment granules. They are the accumulation of material that is not metabolically active. They are usually present in the cytosol only temporarily and they are non-soluble molecules. On the basis of synthesis and uses, these substances or the inclusions are of following types. Like first one, it's secretory inclusion. Secretory substances or secretory inclusions include enzymes, nectar. Actually, some of the substances synthesize such substances which are not concerned directly with nutrition and growth. These are termed as secretory substances, and I have already told you enzymes and nectar come under the secretory substances. Now next we are going to study about nutritive inclusions. It include glycogen, lipid granules scattered in the cytoplasm. It also include pigment granules, crystalline inclusion. Some of the excretory substances are also released like alkaloids, resin, gum, tannin, latex. Mineral crystals in the protoplasm are made up of calcium carbonate, calcium oxalate and silica. So this is all about the non-living inclusions of the protoplasm. Coming back to our topic cytoplasm, it's semi-fluid, jelly-like, translucent, little viscous part of the cell. Contains almost 80% water and 20% portion is responsible for its viscosity, its jelly-like nature. It includes cytoplasmic matrix, cytoplasmic organelles and cell inclusions. We have already discussed about cell inclusion in detail. Now we are going to discuss the functions of cytoplasm or the characteristics of cytoplasm. Actually, it aids in the movement of genetic material and products of cellular respiration within the cell. It helps in cellular respiration with the help of cytoplasmic inclusions. It helps to fill out the cell and keep organelles in their place. As you know that cell organelles are embedded or scattered in the cytoplasm. It gives cell its shape. ये cell को shape भी देता है. It adds to dissolve waste product. Waste products are also dissolved in it. Waste materials are actually formed during metabolism. They are temporarily stored in the cytosol before being eliminated, as we have discussed the process of exocytosis. Through the process of exocytosis, this waste product will be eliminated from the cell. It also stores raw materials for its metabolism as well as functioning of different organelles. Cytoplasm matrix contain enzymes for the synthesis of fat, several carbohydrates, nucleotides, proteins, coenzymes. It's responsible for the exchange of materials. We are going to discuss about cytoplasmic streaming or cyclosis. Cytoplasm allows movement of substances within the cell by a process termed as cytoplasmic streaming. Actually, cytoplasm is not static. But it's always in motion. Movement of cytoplasm within the cell is termed as cyclosis or cytoplasmic streaming. इसका ये मतलब नहीं है कि सेल मूव करेगा. एक्चुअली साइटोप्लाज्म की मूवमेंट हो रही है जो कि कहाँ प्रेजेंट है, एक सेल में, एंड इट्स द फ्लूइड विच इज मूविंग, that is cytosol, not the organelles. But as organelles, you know, are embedded in it, in cytoplasm, so when the fluid is moving, organelles also appears to move, as you can see here. Now what is the utility of this movement? Actually, this movement or streaming of cytoplasm will be helpful in transportation of nutrients and metabolites. So when fluid moves, it helps in it exchange of materials between cell organelles. Hence, it will also lead to cell interactions. Hence, cytoplasm is of fluid nature and it's capable of movement. It can also change its shape. Now, we have discussed about cell boundaries, cell wall, cell membrane, glycocalyx. Today we started cytoplasm, where we read about cytosol. We also read about trophoplasm, which is formed of cell organelles and non-living cell inclusions. We have read the inclusion in details. Now we are going to start cell organelles. So what are cell organelles? As I have already told you, these are permanently metabolically active and living structures of cytoplasm. They are suspended in cytoplasm. Each organelle is surrounded by its own individual membrane. They have distinct form, specific chemical composition, definite function, without which the cell cannot function as whole. Some of the cell organelles, as you can see in the picture, are mentioned: nuclear, endoplasmic reticulum, Golgi body, ribosome, Golgi apparatus, lysosome, ribosome, centrosome. Now what is endomembrane system? Actually, the functioning of certain membrane-bound cytoplasmic organelles is well coordinated. They exchange materials among themselves by means of vesicles. These organelles collectively form endomembrane system. Roots, plasma membrane, endoplasmic reticulum, Golgi complex, lysosomes, vacuoles. Functions of these cell organelles are well coordinated, whereas functions of other cytoplasmic organelles is not coordinated. Hence, rest of the cell organelles are not considered as part of endomembrane system. Now we are starting our first cell organelle or the first member of endomembrane system, that is endoplasmic reticulum. In short, we say it as ER. Cytoplasm of eukaryotic cells contain a network of interconnecting membranes. This extensive structure is called endoplasmic reticulum. Actually, its name suggests endoplasmic reticulum. As its name suggests, endoplasmic reticulum. Endo means inside, plasmic related to plasma or cytoplasm, and reticulum means network. So basically it's a network of interconnecting membranes. is of two types, RER and SCR, that is rough endoplasmic reticulum and smooth endoplasmic reticulum. Due to presence of ribosomes on the surface, this rough endoplasmic reticulum, they have the presence of ribosomes on its surface. That's why their surface look like granular or rough in appearance, and it is termed as rough endoplasmic reticulum or RER. Whereas in case of smooth endoplasmic reticulum, ribosomes are not attached to the surface. That's why the surface seems to be smooth in appearance, and they are known as smooth endoplasmic reticulum. Now, what are the main function of ribosome? Ribosome actually involved in the protein synthesis. So the main function of RER is to help make proteins, and function of SCR is fat or lipid into detoxification. We will discuss their function in detail. They may be tubular, round, oval in shape. They may be of different shapes in different cells depending upon the functions they perform. Now here are the components of ER: cisternae, vesicle and tubules. Cisternae are the long, flattened and unbranched units arranged in the form of stacks, and vesicles, these are oval membrane bound structure. Tubules, these are irregular, often branched tubes bounded by a membrane. Here you can see ER is related close to the nucleus. This is rough endoplasmic reticulum. This dot like structure which you can see is actually ribosomes. They are attached to the surface of RER. And see here, SCR, their surface is free of ribosome, that is ribosomes are not bound here, not attached here. This is SCR. RER continues with SCR on the one side, whereas it's also connected with the nuclear membrane. This is the nucleus, nuclear involved, this is nuclear membrane. So at one side RER is continued with SCR, and other side it's connected to nuclear membrane. And SCR, its first side, one side is continued to the RER and other side is attached to Golgi body. This is the Golgi apparatus or Golgi body, and here SCR is attached with the Golgi. Now we are going to discuss some of the functions of RER and SCR. Firstly, SER. As I have told you, SCR, it's involved in the synthesis, transport as well as storage of lipids. It is involved in the metabolism of carbohydrate, detoxification of drugs, alcohol and poisons. In liver, actually harmful chemicals, drugs, alcohols, poisons, these are detoxified by the liver cell, and for this SCR is present there. And what actually SCR did, it increased the surface area, it doubles the number and increase the surface area so that it can fasten the process of detoxification, and later on it comes to its original number. Now functions of RER, it's involved in synthesis, transport as well as storage of proteins. Proteins का synthesis भी करेगा, transportation भी and storage भी. Proteins are manufactured in ribosome and they are then transported to other parts through endoplasmic reticulum. As you know, RER is synthesizing the proteins, whereas SCR synthesizing lipids, and both these components, proteins and lipids, are involved in the formation of plasma membrane. We are discussing some of the common functions of RER and SCR. They give mechanical support to the cytoplasm. Actually, microfilaments, microtubules and ER, they form the endoskeleton of cell. That's why they provide the mechanical support. Next function is, it is involved in intercellular exchange. Actually, the exchange of molecules by the process of osmosis, diffusion and active transport. ER forms intercellular conducting system, and it's responsible for transportation of material in cytoplasm from one place to another, and also from, as you know, it's connected to plasma membrane, so it's also involved in the secretion of material outside the cell. Next function, it's involved in cellular metabolism. Actually, the membrane of reticulum provide increased surface area for metabolic activities within the cytoplasm. It secretes many enzymes which are involved in various metabolic activities. So these are some of the common functions of RER and SER. This is all about ER. In the next video we will discuss about second member of endomembrane system, that is Golgi complex.